Welcome to my podcast, Scientist Free from Religion. This podcast has as proposal, as main idea to talk about a scientific topic, but also talk about religion. Different from the common assumption in which science and religion should be separated, here on this podcast we recognize formally that science and religion are not disconnected. Sadly, religion can be a very big threat to science. So on this podcast, the proposal is to talk about it freely, so without fear, without the illusion that they are separate topics. So we are going to cover a possible topic. Most of them will be focused on the connection between religion and science, especially to defend science. That's our goal. Our goal here is to defend science from religion and to help to make science, to spread scientific thinking, especially to defend it against religion.
Welcome to The Deep Dive, the show that converts towering stacks of research into concentrated, actionable knowledge. Today we're tackling one of science's most enduring and provocative puzzles. What is life? And maybe more importantly, what kind of scientific explanation for life can actually satisfy us, the human mind?
It's definitely a deep dive into historical biology today, but the core conflict, it feels surprisingly modern. We're tracing this century-long, sometimes really quite furious philosophical battle.
Okay.
On one side, you had people who believed life required some kind of mysterious, non-physical, vital principle.
Right, the old idea of a life force.
Exactly. And on the other side, those who fiercely insisted that life could and absolutely must be fully explained, maybe even synthesized, using just standard physical and chemical laws.
We're pulling from some really rich historical accounts of biological explanation here. And we're focusing on how different scientific cultures sort of defined what knowledge even meant. The sources show us how these early physical models of life often failed, sometimes quite spectacularly. And they got replaced by powerful computational metaphors, think genetic programs, and eventually by new technology that's really blurring the lines between organism and machine today.
And what's really relevant for you, the listener, is that this history isn't just about, debunking old theories like vitalism.
No, it's more fundamental.
It shows us how our cultural needs, our cognitive limits, they dictate what we accept as a satisfying explanation, whether we're talking about how an embryo develops or how a complex gene network functions.
Yeah, that underlying tension, between wanting a simple, neat, maybe mathematical explanation versus accepting the messy, detailed material reality that's really the engine driving our discussion today.
Absolutely. And this whole historical trajectory, it represents a pretty fundamental shift in how we understand existence. It's a move away from explanations that were traditionally dominated by, let's say, non-physical beliefs.
Like vitalism.
Right. Towards more rational, evidence-based, secular inquiry into the actual mechanisms of the natural world.
Okay, let's unpack this conflict then. Let's start at the turn of the 20th century. Because for centuries before that, nobody really felt the need for a super strict definition of life, did they?
Not really. As the sources point out, life was just, manifest, obvious.
You knew it when you saw it. A person, a plant, a cat.
Exactly. But then biology started trying to establish itself as a rigorous science, distinct from chemistry and physics.
Right.
And that boundary, that line between living and non-living, suddenly became this massive problem. It needed defining.
Right, so the demarcation of biology itself kind of forced the issue.
It exacerbated the need, yeah. The question, what is life? It stopped being just a philosophical thought experiment.
That became
a scientific provocation. It demanded an objective scientific solution. And for many scientists back then, that solution felt intrinsically tied to the question of origins, abiogenesis.
You mean how life first arose from non-life?
Precisely. The thinking was, if we could create life from non-living stuff, then we'd finally understand it.
And that feeling led to some pretty wild experiments, didn't it?
Oh, absolutely radical experiments. Which brings us neatly to Stephen Leduc.
Leduc.
Right.
Early 1900s. He was basically doing what he called synthetic biology over a century ago. Amazing.
His goal was nothing short of revolutionary for the time. He wanted to close that perceived gap, that chasm between the living and the non-living, by reconstructing biological phenomena using purely physical means. Specifically, he focused on osmotic pressure.
Osmotic pressure, like water moving across membranes.
Exactly. Leduc had this idea about how science works. He thought it usually goes from observation to decomposition, breaking things down.
Analysis.
Right. And then finally to synthesis, building things back up. But Leduc, he decided to kind of skip the messy decomposition part.
And jump straight to
Synthesis. Let's just build it.
So he wasn't just watching life. He was trying to actually grow something lifelike in a beaker using simple inorganic chemicals.
That was the aim.
What did these famous osmotic growths actually look like? Why were they so convincing to some people?
Well, the process was quite simple. You'd drop specific mineral salts, like potassium ferrocyanide or copper sulfate was a common one, into a solution, often sodium silicate or gelatin and water.
Okay.
When these salts dissolved, they reacted chemically with the solution around them, and this reaction created a kind of skin, a semi-permeable membrane.
Ah, the osmosis part.
Precisely. Water from the surrounding solution would then rush into this little chemical seed because of the concentration difference that's osmosis. This influx of water forces the membrane to stretch, to expand, to grow outwards.
Into shapes.
Into incredible shapes. These chemical precipitates, these growths, were just visually compelling. They formed stalks, spheres, intricate branching patterns.
Like plants or fungi?
Exactly. Visually, they were strongly reminiscent of algae or maybe rudimentary corals or plant structures, and they seem to exhibit 4 key properties people associated with living organisms.
Okay, let's hear them. What were these 4 properties?
Right. First, growth and reproduction. They clearly absorbed material from the surrounding medium. They swelled, they grew larger, often forming these quite complex structures. Some even seemed to bud off smaller structures, mimicking reproduction.
Okay, growth and pseudo-reproduction. What else?
Second, assimilation and elimination. They appeared to take in matter from the solution, chemically transform it within their structure, incorporating it into the membrane, and sometimes they'd even eject waste products. Little bits would break off, or substances would diffuse out through ruptures.
Like eating and excreting in a very basic chemical sense.
Kind of, yeah. And third, and maybe this was the most visually striking, morphogenesis.
Form generation.
Yes, they generated specific characteristic forms. They didn't just grow randomly. They often seemed organized, complex, and sometimes they'd even stop growing once they reached a particular shape, as if self-limiting.
Okay, I can see it now. If you're looking at these things in a dish and they seem to grow, take stuff in, put stuff out, and develop complex, non-random shapes.
Yeah, it's easy to see why Leduc reached the conclusion he did. That osmosis wasn't just a factor, but maybe the predominant physical mechanism behind the origin and continuation of life on Earth. He thought he'd found the fundamental physical basis for life's essential properties right there in his chemical garden.
Wow. So for the physicalist camp, the people arguing against needing a special vital principle.
Leduc's work was huge. It was like Exhibit A.
Tangible proof.
It certainly seemed like it. Tangible proof that this supposed gap between the inorganic world and the organic world was maybe just an illusion. His experiments, even if they seem crude to us now,
Right.
they were hailed at the time as a potential bridge between the province of inert matter and that of living matter. It suggested you didn't need any mysterious vital force to get the appearance of lifelike organization.
But that's the key word, isn't it? Appearance.
Absolutely crucial.
Because Benjamin C. Gruenberg, a biologist writing just a few years later, in 1911, he immediately drew a line in the sand. He made a distinction that really undercut Leduc's big claims.
Gruenberg's critique was sharp and deeply clarifying for the field. He basically said, hold on a minute, there's a massive difference between the synthesis of artificial life, meaning creating convincing lifelike models or simulations.
Like Leduc did.
Right, and the artificial synthesis of life, which means creating actual living matter, something that can truly sustain itself, metabolize robustly, reproduce reliably, evolve.
He was accusing Leduc of confusing resemblance with reality.
Essentially, yes. Conflating a clever chemical mimicry with the actual foundation of life.
It's like the difference between building a hyper-realistic robot that walks and talks.
And creating a genuinely living organism that grows and evolves all by itself, worlds apart.
Did Gruenberg see any value in Leduc's work?
He did, actually. He wasn't completely dismissive. He granted that Leduc's experiments were useful for clearing the field of much conjectural rubbish.
Yes, so.
By showing very visually how purely physical forces like osmosis could spontaneously organize matter into complex shapes. It helped push back against the most naive forms of vitalism.
Yeah.
But for biologists like Gruenberg, who are grappling daily with the staggering complexity of actual cells, the metabolism, the regulation, the genetics, Leduc's simple chemical gardens just seemed, well, simplistic, fragile. They didn't capture the robustness, the self-maintenance, the sheer contingent complexity of real living systems.
So the critique was really about the gap between the model and the messy reality.
Exactly. It refocused the effort. It said, okay, mimicking appearances is interesting, but the real challenge is understanding the underlying molecular machinery and the principles of biological organization. That critique was vital.
Okay, so Leduc's attempt at pure chemical mimicry, it ultimately failed to convince the biologists because it just lacked that deep material complexity.
Right. It scratched the surface, but didn't capture the substance.
But the failure of those early physical models didn't stop others, particularly those with a more mathematical mindset, from trying a different tack.
No, the quest continued.
Instead of mimicking life's substance, they tried to explain life's form using mathematical laws. And this is where the conflict shifts, isn't it? It moves away from vitalism versus physicalism.
And becomes more of an internal dispute within biology itself. A dispute about method, about what counts as a good explanation. The sources talk about a deep epistemological divide.
Epistemological divide. Okay, break that down.
This really gets to the heart of Keller's thesis in the source material. What we accept as a satisfying explanation isn't some universal, fixed thing. It's contingent. It's shaped by what she calls epistemological culture.
Meaning?
The norms, the shared beliefs, the sort of unspoken rules within a particular scientific subculture about what constitutes proper evidence, what counts as real understanding.
So different tribes within science have different standards.
Exactly. And throughout the 20th century, there was this persistent, often quite bitter communication gap between experimental biologists, the ones getting their hands dirty in the lab, focusing on specific, verifiable material facts.
Empiricists.
Right. And the theoretical or mathematical biologists who tended to favor abstract principles, mathematical elegance, and simplified models, they just had fundamentally different rules of evidence.
Okay, let's make this concrete. Darcy Thompson. His book, On Growth and Form, first published way back in 1917, it's considered a classic now, right? A masterpiece of scientific writing.
Absolutely beautiful book. Visually stunning, conceptually profound.
Yet for much of his career, Thompson was kind of on the margins of mainstream biology. Why was that?
Well, Thompson in some ways was continuing Leduc's project, but with mathematics instead of just chemistry. His mission was to explain biological form, why organisms have the shapes they do, and biological growth, using mechanical forces and mathematical principles.
He saw physics as the ultimate benchmark.
He saw the methods of mathematical physics as the conceptual model for biology. He used mathematical analogies, drawing inspiration from simple physical systems, things like the shapes of splashes, the patterns as stress materials, soap bubbles, even Leduc's chemical growths.
To argue what?
To argue that purely physical forces, acting under normal earthly conditions, could spontaneously generate structures very similar to those found in living organisms.
His logic was pretty straightforward then. If we can explain the shape of a soap bubble using math and physics,
then we should be able to explain the shape of a cell, or a jellyfish, or a bone using the same kind of mathematical description. The models weren't meant to be literal copies, but intellectual tools. Prosthetic links, as Keller calls them, in a chain of argument showing that physics was sufficient.
makes sense. But where did he clash with the biologists?
Hey there, thanks for being around. We are going to make a small break. I'm going to share with you my books. I have several books. So far, all my books about religion, atheism, which have been published a lot, they are in Portuguese, which is my main focus. But if you are interested, you can still buy my books to help me to support my work. I have several books. For example, one book that I'd like to recommend now is AI by Sci-Fi. AI by Sci-Fi is a book that talks about artificial intelligence, which is everyone is talking about artificial intelligence with ChatGPT, all this amazing stuff that they can do. They can talk, they can create video, they can create podcasts. This podcast, for example, There are several episodes that was recorded using artificial intelligence, which is amazing how I can do that. I can even interact as you could see in several episodes. So I would like to invite you to buy my book AI by Sci-Fi. It's available on Google Books, Google Play. It's also available on Amazon. So you can buy my book. If you are interested in something a little bit more complex, a little bit more technical, I do have as well a technical book. For example, I have one book about computational thinking, in which I talk about Kasparov. It's a technical book. Greg Kasparov was the first guy to be beaten by a computer. He has a very nice book about that, how artificial intelligence overturned the difference between human and artificial intelligence. So please buy my book and offer me all the support. You can find all of them on Amazon. Hopefully in the future I'm going to write also about religion, atheism and science. But I do have a podcast, I do have a blog called the Scientists Free from Religion, the same name of the podcast, and you can find several already publications about several topics in which talk about science, religion and atheism. So hope you enjoyed the show. So let's come back to the show.
Well, Thomson committed what, in the eyes of most experimental biologists of the time, was a cardinal sin.
Which was?
He essentially dismissed genetics. He openly disregarded the burgeoning field focusing on genes and heredity.
Wait, seriously? In the era when genetics was taking off?
Yep. He saw the focus on this hereditary substance as a kind of distraction. He argued that the dynamic forces, the physical actions happening in the cell and tissues, were far more important for determining form than the specific material stuff passed down in the nucleus.
Wow. So he basically told the people, meticulously mapping genes and mutations, that they were looking at the wrong level, that they were lost in the details.
That's pretty much it. He actually advocated for what he called a principle of negligibility.
Negligibility.
Yeah. He argued that to find the underlying mathematical regularities, the elegant laws of form, scientists must simplify. They must eliminate what he saw as accidental individual differences.
Oh, that sounds dismissive.
It was entirely alien to the whole ethos of experimental biology, especially fields like genetics and embryology. Those researchers often built their entire careers on documenting, exploiting, and tracing those very individual specific differences, a particular mutation, the lineage of a single cell.
They saw the complexity as the reality, not as noise to be averaged out.
Exactly. They believed life was inherently complex and specific, and the goal was to unravel that specificity, not to find some universal equation that smoothed it all away.
I can just imagine the friction. If you're an embryologist who's spent years painstakingly tracking cell movement, and some theorist comes along and says, oh, all that data, that's just accidental noise. We need to neglect it to see the real pattern.
You'd be absolutely furious. It sounds like a fundamental lack of respect for the hard-won empirical facts. And that resentment, that cultural mismatch, is precisely why Thomson's work, despite its brilliance, remained largely an outlier for decades.
Okay, so that cultural clash was already simmering. Then along comes Nicholas Reshevsky.
Reshevsky. A theoretical physicist heavily inspired by Thomson, he had even grander ambitions.
Which were?
To build a complete systematic mathematical biology structured exactly like mathematical physics. A grand unified theory of life based on equations.
Ambitious indeed. He tried to tackle fundamental processes, right? Like cell division.
He did. His major work, Mathematical Biophysics, tried to explain cell division purely as a consequence of physical forces arising from metabolism.
And his model, let me guess, involves some simplification.
Oh, just a bit. Predictably, it centered on an idealized, perfectly spherical cell.
The infamous spherical cow problem in biology.
Exactly that. His model tried to derive the geometry of cell division, why a cell splits in two, purely from the diffusion gradients of nutrients coming in and waste products going out, all within this mathematically convenient, perfect sphere.
Mathematically elegant, I assume?
Brilliant mathematics, apparently. Terrible biology.
So how did the experimental crowd react when Reshevsky presented this at the big Cold Spring Harbor Symposium back in 1934? I imagine it wasn't a warm reception.
Immediate and defining. Biologists like Charles Davenport and the hugely influential E.B. Wilson were in the audience. They acknowledged the math was clever, but they essentially declared the work useless for understanding real biology. Davenport apparently said something like, the explanation for the spherical cell dividing might be very satisfactory mathematically, but it's irrelevant because the spherical cell isn't the commonest form of cell. Real cells are often weird shapes, ovoid, columnar, flat, spiky. Reshevsky was solving a problem that didn't reflect biological reality.
And Reshevsky probably argued, well, you start simple and generalize later, right? The standard physicist's defense.
He did, but that did not fly with the empirical mindset of the biologists.
No.
Absolutely not. E.B. Wilson, who was a giant in the field, basically stood up and laid down the law. He articulated the core beliefs, the fundamental axioms of the biological ethos at the time. These weren't gentle suggestions. They were a manifesto against what they saw as theoretical arrogance.
Okay, what were these axioms or platitudes, as the source calls them?
Wilson was blunt. First, science need not be mathematical, a direct shot.
Wow. Okay.
Second, simply because a subject is mathematical, it need not therefore be scientific, implying that math could be just formal game playing, detached from reality. And perhaps the most cutting one for modelers. Empirical curve fitting may be without other than classificatory significance.
Meaning, just because your equation fits my data points doesn't mean your model actually explains the underlying mechanism. It might just be a fancy description.
Exactly. For Wilson and his colleagues, mathematics was strictly auxiliary. Its role was to help find out the facts and identify the important variables through observation and experiment, not to dictate the explanation from first principles.
So Reshevsky was seen
as arrogant, out of touch with biological complexity. His abstract modeling was judged basically as futile when the actual facts about growth and form were, as biologist Eric Ponder put it around that time, not only very complex but largely unknown.
So that encounter really solidified the mainstream rejection of that kind of top-down, physics-envy theoretical biology for a long time.
For decades, really, the message was clear. Get the facts first, then maybe use math to help make sense of them. Don't start with the math.
Right. So by the mid-1930s, the biologists had firmly pushed back against these overly simplistic physical and mathematical models. If your model ignored the messy details of a living cell, they were going to ignore your model.
Pretty much, yeah. The focus swung heavily towards genetics, towards the material basis of heredity.
But then, almost 2 decades later, in 1952, a very famous name enters the picture, someone completely outside biology at the time, Alan Turing.
The codebreaker, the father of computer science. Yeah, a surprising figure to pop up in developmental biology.
He published this theoretical paper on morphogenesis, the generation of biological form.
He did, the chemical basis of morphogenesis. And conceptually, it was absolutely groundbreaking, even though it also faced a pretty frosty reception initially.
What was his idea? How was it different?
Turing proposed what's called a reaction diffusion model. He showed mathematically how spatial pattern structure could emerge spontaneously from a system that was initially completely uniform, completely homogeneous.
A featureless sphere, as the source puts it.
Exactly. He imagined two hypothetical chemicals, which he called morphogens, form producers reacting with each other and diffusing through the space at different rates.
And this interaction could create patterns from nothing.
That was the key insight. The interplay between the reaction kinetics and the different diffusion rates could destabilize the uniform state and lead to stable, repeating spatial pattern spots, stripes, networks. It solved the chicken and egg problem of development in a theoretical sense.
How so?
You didn't need a pre-existing pattern or blueprint to create the pattern. The pattern emerged from the dynamics of the system itself. It was a self-organizing mechanism.
Which is intellectually very appealing. No need for some mysterious guiding hand or complex pre-patterning.
Exactly. It offered a potential mechanism for complexity arising from simplicity. But, and it's a big but.
Here we go again, simplification.
Massive simplification. Just like Reshevsky, Turing achieved this mathematical elegance by deliberately and drastically simplifying the biological reality.
What did he leave out?
Crucial stuff. He ignored cell structure altogether, treating the embryo basically as a continuous chemical soup. And perhaps most controversially for the time, he explicitly sidelined genes from the core dynamics. He assumed their role was purely catalytic, maybe influencing the rates of the morphogen reactions, but not determining the pattern itself.
Wait, this is 1952. Watson and Crick are just around the corner. DNA is becoming the molecule of life. How could Turing, this genius of computation and logic, think he could sell a model of development to biologists by essentially ignoring the gene.
Well, he was very upfront about it. He explicitly called his approach a simplification and an idealization, and consequently a falsification.
A falsification. He admitted it was wrong.
Wrong as a literal description of an embryo, yes. He saw his model not as the answer, but as a proof of possibility. He was demonstrating that purely physical and chemical dynamics could, in principle, generate biological patterns without needing complex genetic instructions for every detail.
Okay, so it's about shifting the causal explanation.
Precisely. This is a really fascinating point Keller makes. The causal responsibility for the pattern wasn't assigned to some prior agent, like a specific gene being turned on or a specific founder cell dividing.
It was assigned to
The set of interaction dynamics, the self-organizing properties emerging from the reaction and diffusion rates. The cause was in the system's behavior, not localized in a specific component.
I can see why that would be unsatisfying for a biologist focused on finding the specific gene or molecule responsible for a specific trait. It feels diffuse, less concrete.
Deeply unsatisfying for many, it lacked that tangible material cause they were trained to look for, and the reception reflected that.
How so? Was it just ignored?
Largely, yes. For nearly 2 decades, it remained a theoretical curiosity. Even someone like C.H. Waddington, who was actually quite open to theoretical ideas and respected Turing.
Waddington was a major figure, bridging genetics and embryology.
Right. Even Waddington, while praising Turing for bringing competent mathematicians into biology, had some really sharp critiques.
What were his main objections?
His core reservation was fundamental and echoed the critiques of earlier models. He pointed out that the starting point for real development, the egg cell, is very seldom, if ever, a featureless sphere.
It already has structure.
Absolutely. There are often maternal factors, proteins, and mRNAs already laid down in specific locations within the egg cytoplasm before fertilization even happens. There's inherent inhomogeneity from the get-go.
So if Turing's model needed a perfectly uniform starting point to generate a pattern spontaneously,
But the embryo never provides that perfectly uniform starting point.
then the model, however clever mathematically, might be irrelevant for explaining how actual embryos develop their main body plan.
That was Waddington's point. He thought maybe it could apply later to things like pigment patterns on skin leopard spots, zebra stripes, which develop on a relatively uniform surface. But for core embryogenesis, probably not.
Did he have other critiques?
Yeah, he also pointed out that biological patterns often aren't just static things that form once. They often involve active, dynamic equilibrium. Think of a flame, it maintains its shape dynamically. Waddington felt biological patterns were often more like that, actively maintained and self-regulating against perturbations, not just passively formed by diffusion.
So the ultimate reason Turing's model got shelved for so long wasn't just the simplifications.
It was a deeper lack of confidence among the experimental community. They just didn't believe that these kinds of highly abstract, theoretically imagined accounts based on hypothetical morphogens with unknown reaction rates could actually satisfy their need for explanation. They needed empirical grounding.
It comes back to that quote from Eric Ponder again, doesn't it? It is futile to conjure up in the imagination a system of differential equations for the purpose of accounting for facts, which are not only very complex, but largely unknown.
Exactly. The feeling was, show us the molecules, measure the rates, ground the theory in empirical reality. Until then, it's just an interesting mathematical exercise, not real biological explanation. The empirical facts had to come first.
Okay, so Leduc's chemical gardens, Thomson's geometry, Reshevsky's spherical cells, even Turing's brilliant reaction diffusion. None of these early physical or mathematical models really managed to become the dominant way biologists explain life in the mid-20th century.
No, they remain on the sidelines for the most part.
Instead, the focus shifted, laser-like, onto the gene.
All eyes on the gene.
But here's the twist. Even the gene itself needed a conceptual makeover to really provide a satisfying, comprehensive explanation for the complexities of life, didn't it?
It absolutely did. And that makeover was powered largely by metaphor, powerful, compelling metaphors.
Let's talk about the gene before DNA. In classical genetics, what was the gene?
It was a fascinating entity, functionally essential but physically mysterious. Keller calls it an abstract, multivocable entity, meaning it could mean slightly different things depending on the context. It was a kind of a real fiction.
A real fiction? How so?
It was real because geneticists could map its location on chromosomes, track its inheritance, predict its effects. But it was fictional in the sense that nobody knew its physical nature. Geneticists use the concept of gene action. The gene does this, the gene controls that, as a kind of placeholder. It provided a provisional causal narrative for development and heredity, allowing the field to progress even with this big black box at its center. It kept the internal incoherence under wraps, as Keller puts it.
Okay, so it was a useful abstraction, but then, boom, the double helix, 1953.
Right. The molecular revolution hits. Suddenly, the gene isn't an abstract placeholder anymore. It acquires a concrete physical chemical identity. It's a specific sequence of bases on a molecule of DNA.
Problem solved.
Well, it solved the chemical mystery, but it created a new conceptual problem. DNA, fundamentally, is a relatively inert molecule. It just sits there. How do you get dynamic, purposeful biological action development, regulation, behavior out of this static chemical string? Where did the agency go?
The gene action narrative suddenly felt less convincing when the gene was just stuff.
Exactly. You needed a new story. And that new story came brilliantly from the burgeoning field of computer science and information theory.
Enter Jacob and Monod.
Francois Jacob and Jacques Monod, in 1961, studying gene regulation in bacteria, they introduced the concept of the genetic program, and they were explicit about borrowing the metaphor from computers.
This was the masterstroke.
Absolutely. It was an incredibly powerful metaphor, because the agency, the purposefulness, the sense of direction that the gene had lost when it became just a DNA molecule, it was more than regained through this new language. The gene now became information. It became the program.
So the DNA molecule itself might be passive, but the sequence, the information it contained, was active.
Precisely. The gene was redefined as the master molecule, the director, the holder of the plan, issuing instructions. Other cellular components, proteins, enzymes, were cast in the role of obedient servants executing the program.
That narrative structure, director and servants, must have provided a huge sense of clarity for molecular geneticists trying to make sense of complex processes.
Immense clarity and coherence. It gave them a powerful framework for thinking about gene regulation and development. But it also had a crucial, perhaps less obvious effect.
Which was?
Jacob and Monod effectively tamed the cybernetic metaphor. Cybernetics, which was influential in the 40s and 50s, talked broadly about feedback, regulation, and self-adjusting systems in both machines and organisms.
Okay.
But Jacob and Monod took terms like feedback and regulation and gave them a very specific gene-centric meaning within their operon model. Feedback repression, for instance, referred specifically to a genetically controlled process where the end product of a pathway inhibits the gene responsible for making an enzyme early in the pathway.
So they narrowed the focus.
They narrowed the focus primarily onto regulation by the genes. This framing implicitly downplayed or marginalized other potential regulatory mechanisms happening in the cell, mechanisms that weren't directly encoded in the DNA sequence itself.
Can you give an example of such a mechanism that got sidelined conceptually?
A key one is allosteric inhibition or regulation.
Okay, what's that?
This is where a regulator molecule binds to an enzyme at a site other than its main active site. This binding changes the enzyme shape, either switching it on or switching it off. It's a super important way cells fine-tune metabolic pathways moment to moment without changing which genes are expressed.
So it's regulation, but not directly commanded by the gene sequence.
Exactly. It's inherent in the protein's structure and its interactions with other molecules. But because the genetic program metaphor put the gene firmly in the central processor role, these kinds of non-genetic, dynamic regulatory mechanisms, while certainly known and studied, were often conceptually relegated to a secondary, less fundamental tier, especially when thinking about the big picture of development and differentiation. The powerful program metaphor steered attention towards the master molecule.
Okay, so the program metaphor brought clarity, but also perhaps narrowed the explanatory focus. Now, speaking of simplifying complex realities, there was another hugely influential metaphor, or maybe framework, that emerged around the same time, late 1960s, Lewis Wolpert's concept of positional information.
Ah, Wolpert, yes. Another engineer by training who brought a different kind of thinking to embryology. He was looking for a general, abstract framework to understand pattern formation, how cells in an embryo figure out what they're supposed to become based on where they are.
And his core idea was
deceptively simple, yet profound. He postulated that a cell knows where it is, and this information specifies the nature of its differentiation. He wanted to reduce the mind-boggling complexity of development down to a kind of cellular addressing system.
Like cells having GPS coordinates that tell them whether to become, say, a thumb cell or a pinky cell.
That's the basic idea. Forget the intricate history of cell divisions for a moment. Just focus on the cell's current location within some kind of coordinate system.
And he made this concrete with his famous French flag model, right?
Yes, the French flag model became iconic. It was a simple thought experiment. Imagine a line of cells and imagine a chemical morphogen diffusing from one end, creating a concentration gradient high at one end, low at the other.
Okay.
Wolpert suggested that cells could read this concentration. If the concentration was above a certain threshold, they differentiate into blue cells. If it was between two thresholds, they become white. And if it was below the lower threshold, they become red. Voila, a French flag pattern.
And crucially, the pattern forms correctly, regardless of the total number of the total length of the line.
Exactly. It depended only on the relative concentration, the position within the gradient, not the absolute size. This addressed a key problem in development called size regulation.
A really elegant simplification. But I remember reading that Wolpert faced some pretty intense pushback initially. Why?
Again, it came down to that clash of cultures. The sheer simplicity of the French flag model felt almost insulting to many developmental biologists who were immersed in the incredible, intricate details of cell signaling pathways, gene interactions, and morphogenetic movements.
Too abstract, too detached from the molecular nuts and bolts.
Precisely. Wolpert himself recalled biologists confronting him, sometimes quite aggressively asking, "Who do you think you are?" Presenting such a stripped-down scheme for something as complex as embryonic development seemed naive or even arrogant to those deep in the experimental trenches.
But the idea stuck around, didn't it?
It proved incredibly fruitful despite the initial hostility. Why? Because it provided a unifying, abstract, conceptual space. As molecular biology started generating vast amounts of data about genes involved in development, the positional information framework offered a way to organize that data, to think about how gene activity might relate to spatial position, and to design experiments to test those relationships. It gave people a way to think about the forest, not just the individual trees.
So we've journeyed from failed physical models like Leduc's gardens through the dominance of computational metaphors like the genetic program and positional information. Now, the story takes another turn, doesn't it? As we get towards the end of the 20th century and into the 21st, new technologies start to fundamentally rewrite the rules again.
Absolutely. We see a remarkable convergence happening, bringing together the theoretical, the visual, and the hardcore empirical in new ways.
And perhaps the most dramatic change driver has been our ability to actually see life happening at the molecular level.
Without a doubt, the revolution in biological visualization has been transformative. Things like green fluorescent protein, GFP.
Allowing you to tag specific proteins and watch them move in living cells.
Exactly. And combined with advances like confocal microscopy, which gives you sharp optical sections, and techniques like video-enhanced contrast, these tools have finally broken a really fundamental historical barrier.
Which was?
The incompatibility between visibility and vitality. For most of biological history, if you wanted to see the fine details, the microstructure inside a cell using electron microscopes or staining techniques, you had to kill the cell. You had to fix it, preserve it.
So you had a static snapshot, but you lost the dynamics to life.
Precisely. You were always choosing see the structure or watch the living process from the outside. But now, that choice is often gone. Researchers can literally watch in time. They can see molecules trafficking, cytoskeletons rearranging, cells dividing, all unfolding inside living cells in real time.
That must have been mind-blowing for scientists used to static images.
The sources describe it as an unexpected miracle. It's profoundly changed the way many biologists think and work. It's revitalized that very old scientific tradition that direct observation is the most reliable, most satisfying source of knowledge.
So for many, seeing it happen is understanding it.
To a significant extent, yes. As Keller notes, for many researchers today, seeing is itself a kind of understanding. Watching the machinery in action can feel so viscerally explanatory that the need for further, more abstract theoretical explanation sometimes diminishes.
But this new power visualization didn't just make theory obsolete, did it? In some cases, it actually validated some of those abstract theoretical ideas that had been dismissed decades earlier.
This is the beautiful irony, the convergence. It led directly to the vindication of the morphogen concept.
Ah, Turing and Wolpert's hypothetical form-generating chemical.
Exactly. The absolute classic example is the work on the fruit fly embryo Drosophila, particularly the bicoid protein.
What did they find?
In 1988, Christian Nusslein-Volhard and Wolfgang Driver did this landmark work. They showed that the bicoid protein, which comes from mRNA deposited by the mother at one end of the egg, forms a perfect concentration gradient across the length of the early embryo. High at the head end, low at the tail end.
So a real molecular gradient, not just a hypothetical one.
A real, measurable molecular gradient. And it functioned exactly as Wolpert's positional information model predicted. Different concentrations of bicoid protein activate different downstream genes, telling cells where they were along the head-to-tail axis. Wolpert himself called it the clearest demonstration of a positional signal.
And how was this gradient actually maintained? Was it simple diffusion?
It turned out to be a dynamic balance, exactly the kind of thing Turing's math explored. It involved diffusion, the bicoid protein spreading out from its source combined with degradation, the protein being constantly broken down throughout the embryo. This dynamic interplay created and maintained the stable exponential gradient.
So the very mechanisms Turing had explored mathematically decades earlier were actually at play.
Precisely. This work was huge because it provided concrete molecular proof for the morphogen concept. It rehabilitated that once disreputable term. Suddenly, diffusion gradients weren't just abstract theory, they were measurable biological realities. It beautifully bridged the gap between the molecular geneticists who had the molecules and the theorists who had the concepts. The math had pointed in the right direction after all.
Okay, so visualization confirms theory. And as the molecular details of genes became clearer, did the genetic program metaphor also evolve?
It did. It started to shift from just a metaphor towards something more literal, especially as scientists began to grapple with the complexity of gene regulation. It wasn't just about a master molecule giving orders anymore.
It became more about information processing.
Exactly. The idea of the genetic computer started to take hold. Eric Davidson's work on the sea urchin embryo, specifically the regulatory region, the promoter of a gene called endo-16, is a fantastic case study here.
What was special about that promoter?
Its sheer complexity. This one stretch of DNA that controls a single gene had something like 50 different binding sites for various transcription factor proteins, the molecules that actually turn genes on or off.
50 inputs for one gene. How do you even begin to figure out how that works?
You can't really just by doing traditional wet lab experiments one factor at a time. The complexity is overwhelming. Davidson realized this required a completely different approach.
Which was?
A massive iterative back and forth between computation and wet lab experiments. His team built a quantitative computational model of the endo-16 promoter, trying to capture how it integrates all those different protein inputs. They'd make predictions with the model, then go back to the lab to test them by mutating specific binding sites, then feed the results back into refining the model.
And what did the model reveal?
It showed that the promoter operated in a surprisingly logical manner. Different modules within the promoter process specific combinations of inputs, almost like subroutines, before integrating them to produce a final output, turn the gene on at the right time, in the right place, at the right level. One researcher, Gregory Ray, described it as acting like a logic circuit or a central processor.
So the computer analogy wasn't just loose talk anymore. The promoter was literally computing.
It was performing computation, integrating multiple inputs to make a decision. The model wasn't just descriptive, it was predictive. It became a guide for actually intervening, for modifying the circuitry, for engineering the system.
This really does sound like the line between explaining life and engineering life is getting incredibly thin. The genetic computer is becoming less a metaphor and more literal.
It approaches literality, especially now with the rise of synthetic biology, where people are designing and building genetic circuits from scratch. But maybe the most profound insight to come out of this kind of computational modeling wasn't just about logic, but about a property often taken for granted in biology: robustness.
Robustness. What do you mean by that in this context?
Biological systems are incredibly reliable, aren't they? They manage to develop and function correctly despite all sorts of slop and noise. Think about an embryo developing. It usually turns out okay, even if the temperature fluctuates a bit, or if there are minor genetic variations between individuals, or if the number of signaling molecules isn't exactly perfect every time.
Right, they buffer against variations. They're not fragile.
Exactly, they're robust. Computational models, like one developed by Von Dasso and Odell for the segment polarity network in Drosophila, another key patterning system, started focusing specifically on this property. How do these gene networks achieve such stability?
And what did the modeling reveal? Something unexpected.
Totally unexpected, according to the researchers. They found, through exploring the space of possible network parameters computationally, that networks capable of producing the observed pattern reliably, networks that were robust to variations in parameters, were actually quite rare in the theoretical space of all possible networks.
So being robust isn't easy.
It seems not. This led them to a really profound hypothesis. Maybe robust gene networks aren't just a nice feature of biological systems. Maybe they are effectively the only kinds of networks that natural selection can successfully evolve and maintain over long timescales. Less robust solutions might arise, but they'd be too fragile to persist.
Wow, that's something you might completely miss if you were just doing traditional experiments, right? You'd observe the robustness, but maybe just assume it's there.
Exactly. The computational modeling highlighted robustness not just as a feature, but as a fundamental constraint on biological evolution, shaping the very logic of life. It shifted the explanatory focus. It's not just about how the pattern forms, but how it's formed reliably and stably against the inevitable noise of the real world.
So it really feels like we've come full circle in a way. From Leduc trying to build fragile chemical analogies in a beaker.
Right, focus purely on appearance.
To scientists now using incredibly powerful computational models, tightly integrated with experiments and visualization, to decipher the complex logic, the dynamics, and even the evolved constraints of actual living systems. It confirms, in a way, that life can be explained, measured, and maybe even engineered using these secular, physical, informational, computational laws.
And the explanation that satisfies us now isn't simple. It's not one equation or one molecule. It's complex, multi-layered, involving dynamics, information, computation, and direct observation. It requires embracing that complexity. #tag outro.
Okay, so this deep dive, it's really shown us that the long victory over the idea of a separate vital principle wasn't straightforward, was it?
Not at all. It was messy, full of detours.
It wasn't won by a single perfect physical explanation suddenly appearing. It was more like this slow accumulation, failed models teaching us something, powerful metaphors giving us ways to think, and then finally, technology allowing us to see and compute in ways that validated some ideas and revealed deeper principles like robustness.
And it underlines that core point. What actually counts as a satisfying scientific explanation is fundamentally, well, unstable. It shifts. It depends on the tools we have, the culture of the scientific field we're in, and maybe even our own cognitive needs and limits.
Indeed. We started off asking what constitutes a sufficient explanation for life. And what we've seen is that our own human cognitive capacity is, let's face it, finite.
Limited.
The sheer staggering complexity of something like embryonic development might simply be too vast, too interconnected to ever be fully grasped or held in its entirety within a single human mind.
That seems increasingly plausible. A complete reductionist catalog of every molecule's position and every reaction rate might be theoretically possible someday, but practically useless for genuine understanding. It would be too much data, not enough insight.
Which raises that critical point again. If we can't grasp the whole complexity, we have to simplify. We need models, metaphors, frameworks.
Yeah.
But how do we simplify without falling into Darcy Thompson's trap of just dismissing crucial details as noise?
Yeah, how do we simplify without oversimplifying?
What does this history suggest?
I think it strongly suggests we have to embrace explanatory pluralism. We can't afford to be dogmatic about one right way to explain life.
Meaning we need all the approaches?
We need them all, working together. We need Thompson's appreciation for physical forces and mathematical form. We need Wolpert's abstract frameworks for thinking about organization and information. We need Jacob and Monod's powerful metaphors for agency and program. We need Davidson's computational logic. And underpinning it all, we need the molecular and cell biologists' unwavering commitment to the empirical facts, to the messy, specific material reality that we can now visualize with such incredible clarity.
So instead of searching for that single, elusive, unified theory of life.
Maybe our best chance of truly coming to terms with the living world around us lies in respecting and skillfully utilizing the diversity of all these different ways of knowing. We need the models, the metaphors, and the direct sight.
So what does this all mean? It means perhaps that the greatest tool we possess for understanding life isn't a single theory or a single technology. It's the combined power of diverse human ingenuity, deployed collaboratively to describe, model, and make sense of a reality that might just be fundamentally more complex than any single viewpoint can ever fully capture.
Precisely. A humbling but also empowering thought. Thanks for joining us for the deep dive.
Hey there, you arrived to the end of this episode. You can always check out my YouTube channel and also keep watching because there are 7 episodes. So you just if you enjoyed this one, hopefully you are going to enjoy the next one. I always like to remember that you can support my work by buying my books. Even though I still don't have a book about religion, which is the main topic of the podcast, I do have a book about artificial intelligence, and also I have a blog called Scientists Free from Religion on Medium. And there are several blog posts there, and you can enjoy it, you can read it, and you can subscribe to my YouTube channel, you can share it with your friends. So hopefully I can hear from you and hope you had an amazing time. So hope to see you in the next episode.